Welcome to the Cognitive Neuroscience series. In this series, we will cover neuroscience from a cognitive science point of view. In this video, we will discuss vision and perception. We will start from the most dominant sensory system in the vision and move on to answer how sensation is transmitted to perception. Like most other diurnal creatures, humans depending on our sense of vision. Although other senses such as hearing and touch are important, visual information dominates our perceptions and appears to frame how we think. Both audition and vision are important for perceiving information at a distance, engaging in what is called remote sensing or exteroceptive perception. The advantages of remote sensing are obvious. An organism surely can avoid a predator better when it can detect the predator at a distance. Visual information is contained in the light reflected from objects. We need sensory detectors that respond to the reflected light to perceive objects. As light passes through the eye's lens, the image is inverted and focused to project onto the retina, which is made up of densely packed layers of neurons. The deepest layers are composed of millions of photoreceptors that contain photopigment protein molecules that are sensitive to light. Unlike most neurons, the retina's photoreceptors do not fire action potential. Instead, the decomposition of the photopigments alters the membrane potential of the photoreceptors, and the resulting chemical changes trigger action potential in downstream neurons. The photoreceptors consist of rods and cones. Rods contains photopigment rhodopsin destabilized by the low light levels. Therefore, rods are most useful at night when the light energy is reduced. Cones contains a different type of photopigment called rhodopsin. Cones require a more intense level of light but can replenish their pigments rapidly. Thus, cones are most active during the daytime vision. There are three types of cones defined by their sensitivity to different regions of the visible spectrum: blue, green, and red spectrum. The activity of these three different receptors ultimately leads to our ability to see colors. Rods and cones are not distributed equally across the retina. Cones are densely packed near the center of retina, in the region called fovea. Few cones are in the more peripheral regions of the retina. In contrast, rods are distributed throughout the retina. The rods and cones are connected to bipolar neurons that synapse with a ganglion cells, the output layer of the retina. The axons of these cells form a bundle, the optic nerve, that transmits information to the central nervous system. Before any information is shipped down the optic nerve, however, extensive processing occurs within the retina, where an elaborate convergence of information takes place. The rods can activate a ganglion cell by summing their output even in low light situations. However, each ganglion cell is innervated by only a few cones. These few receptors provide much more detailed information for a smaller region of space, ultimately providing a sharper image. Each optic nerve splits into two parts before entering the brain. The temporal branch continues to transverse along the ipsilateral side, and nasal branch crosses over the project to the contralateral side. The crossover place is called optic chiasm. The pathway that contains more than 90% of the axons in the optic nerve is retinogeniculate pathway, the projection from the retina to the lateral geniculate nucleus of the thalamus. The LGN is made of 6 layers. One type of ganglion cell, the M cell, sends output to the bottom two layers. Another type of ganglion cell, the P cell, projects to the top four layers. The remaining 10% of the optic nerve fibers innervate other subcortical structures, including the purvinar nucleus of the thalamus and the superior colliculus of the midbrain. The superior colliculus and purvinar nucleus play a large role in visual attention. The final projection to the visual cortex is via geniculocortical pathway. This bundle of axons exits the LGN and ascends to the cortex, and almost all of the fibers terminate the primary visual cortex of the occipital lobe. The visual system identifies the what and where of objects. We will look first at the characteristics of the visual neurons and then at the anatomical and functional organization of the visual cortex. Neurons in the visual system keep track of where objects are located in space by responding only when a stimulus is presented in a specific region of space, defined as the receptive field of the neuron. The receptive field of visual cells within neural regions such as the LGN or V1 form an orderly mapping between an external dimension, in this case spatial location, and the neural representation of that dimension. In the vision, these tonotopic representations are referred to as retinotopic maps. A full retinotopic map contains a representation of the entire contralateral hemifield. The receptive field of the cells in the LGN are circular, responding vigorously whenever a stimulus spans a cell's on region but not its off region. These circular receptive fields combine and form oriented receptive fields in V1 neurons. Now, when the cell is active, the signal indicates not only the position of the stimulus, but also the orientation of the edge of the stimulus. The nervous system is integrate in change. For example, we recognize an elephant not by the homogeneous gray surface of its body, but by the contrast of the gray edges of its shape against the background. Neurons that are sensitive to a particular region in space are clustered together. Within this cluster, the orientation tuning of the neurons changes in a continuous manner. Cells also show specifically concerning the source of input. Some cells respond to input from the right eye and the other left eye inputs, forming oculodominant columns. These different properties, location, orientation, source of input, and color can be seen as fundamental building blocks from which percepts are constructed. The optimal stimulus became more complex as information moves through the system. Cells in the retina and LGN respond best to small spots of light, while the cells in V1 are sensitive to edges. Further up system in areas like V4 and TE, the optimal stimulus became much more complex, such as shapes or even faces. Correspondingly, receptive field became larger across the visual system. The boundaries between anatomical adjacent visual areas are determined by topographic reversals. This is the economy of an organization that has been adopted by nature. The visual cortex, where each visual area has its retinotopic map, and borders are defined by such reversals. Now, we will discuss relationship between sensation and perception using vision. But the next video will discuss the details about how we take the information from our sensor system and use to recognize objects and senses. An important question here is that what stage of processing does this sensor stimulation become a percept, something we experience phenomenally? One way to study this question is to trick our sensory processing system with stimuli that cause us to form percepts that do not correspond to the true stimuli in the environment, that is, to perceive illusion. For instance, if we look at the color disk that changes color every second from the red to green, we have no problem seeing the two colors in succession. If the same display flips between two colors 30 times per second, however, the percept is of a fused color, a constant yellowish white disk. This phenomenon is known as a flicker fusion. Okay, then what stage in the visual system does our perception break down, falling to keep up with the flickering stimulus? Does breakdown occur early in the processing within the subcortical structures, or later, or in one of the cortical visual areas? The result of fMRI studies might show us an answer. This study shows that subcortical processing and several of the lower cortical processing areas, V1 and V4, were able to distinguish among the 5 Hz flicker, the 30 Hz flicker, and the 5 Hz non-flickering control. In contrast, the both response within VO, a visual area adjacent to V4, did not differentiate between the high flicker stimulus and the static control stimulus. Here then is the answer. A yellowish object that is not flickering is formed in this higher visual area, known variously as either VO or V8, indicating that although the information is sensed accurately at the earlier stage within the visual system, conscious perception, at least of color, is more closely linked to higher area activity. Therefore, our primary sensory regions provide a representation that is closely linked to the physical stimulus, and our perceptual experience depends more on activity in secondary and associated sensory regions. Okay, this was the vision and perception in a cognitive neuroscience view. The next video will cover object recognition. See you then.